Hi everyone, Dr. Rashid here. In this video, I will be explaining you the metabolism of xenobiotics in detail. So first of all, you should know what is xenobiotics. What is the word meaning of xenobiotics? So the word meaning of xenobiotics is stranger. So xenobiotics means any foreign chemical compound. So it can be any environmental pollutant or any chemical compound present in the processed food items like sweetening agent, coloring agent, or any food preservatives, or it can be any drugs. So any foreign chemical compounds are considered as xenobiotics. So xenobiotics are of two types. One is exogenous xenobiotics, that is chemical compound produced outside the body, or endogenous xenobiotics. These are the chemical compound produced as a result of the intestinal bacterial metabolism. And all of you know regarding the intestinal metabolism of amino acid derivatives. These amino acid derivatives undergo decarboxylation reaction by intestinal bacteria and produces their corresponding amine group. For example, histamine produces histamine, and cadaverine, putrescine, tryptamine, and tryptamine. All these are decarboxylation products which are derived from amino acid. And regarding this metabolism of these xenobiotics, this xenobiotics having one specific feature. All these xenobiotics are lipophilic molecule. Lipophilic molecule. So during this metabolism, these lipophilic molecules are converted into hydrophilic compound. So a compound which should be excreted from our body must be in hydrophilic form. So all these foreign chemical molecules are lipophilic in nature. So during this metabolism, this lipophilic chemical substances are converted into hydrophilic form so that they can be excreted from our body. So before going to the explanation part of the metabolism of xenobiotics, you should know what is biotransformation, what is bioactivation, and what is detoxification. So biotransformation means here a chemical compound is changed from one state to another state. Usually a lipophilic molecule is converted into hydrophilic state. So a lipophilic state, they are converted into hydrophilic state. That is biotransformation. And these biotransformations are of two types. One is toxication, second one is detoxification. So toxication, it is also known as bioactivation. So what is bioactivation? So some chemical compound during their metabolism, they are converted into more toxic form compared to their precursor molecule. That is known as bioactivation or toxication. The classical example is metabolism of vinyl chloride. Vinyl chloride. So vinyl chloride is a chemical compound present in the plastic products. So during the metabolism, this vinyl chloride is converted into their epoxide form. And this epoxide form of vinyl chloride is more toxic than their vinyl chloride form. Moreover, this epoxide form is having carcinogenic effect. So that process or that conversion is known as bioactivation or toxication. Here, a chemical compound after the metabolism, it is converted into more toxic form. And the classical example is vinyl chloride. Vinyl chloride after the metabolism, it is converted into more toxic epoxide form. The second type of biotransformation is detoxification. So the detoxification, as the name means, it is the conversion of a toxic substance into non-toxic or less toxic form. So a toxic substance is converted into non-toxic hydrophilic form. That is known as detoxification reaction. So biotransformation can be either bioactivation or detoxification reaction. Coming to the metabolism of xenobiotic compound. This xenobiotic compound undergo metabolism in two phases. One is phase 1 reaction and second one is phase 2 reaction. And in phase 1 reaction, some functional groups are added to the chemical molecule so that they are converted into phase 1 metabolite. And this phase 1 metabolite undergo further metabolism during their phase 2 reaction. And in phase 2 reaction, these phase 1 metabolites are undergo conjugation reaction with more hydrophilic substances. So these phase 1 metabolites are converted into more hydrophilic, non-toxic compound so that they can be excreted from our body. So regarding phase 1 reaction, in phase 1 reaction, some functional groups are added to the chemical groups. And this phase 1 reaction produces phase 1 metabolite, which are less toxic compared to their precursor molecule. And this phase 1 metabolite undergo further reaction in phase 2. And in phase 2, this phase 1 metabolite undergo conjugation reaction with some hydrophilic compound. So these lipophilic molecules are converted into hydrophilic non-toxic compound. So that's all regarding phase 1 reaction and phase 2 reaction. Coming to the reactions which takes place in phase 1 and phase 2. In phase 1, these are the different types of reaction. The major type of reaction is hydroxylation reaction. Other reactions are oxidation reaction, reduction reaction, and hydrolysis. So these are the different types of reaction takes place in phase 1 metabolism. So phase 1 metabolism is an important short answer question for your university exam. So if the question is phase 1 metabolism, you have to mention about hydroxylation reaction, oxidation, reduction, and hydrolysis reaction with example. So let's see in detail. So the major phase 1 reaction is hydroxylation reaction, and this hydroxylation reaction is mediated by a specific group of enzyme which is known as Cytochrome P450 enzyme system. Actually, it is a group of enzymes which are present in our body. There are around 20 to 200 isoenzyme form of this Cytochrome P450 in our body. And all these Cytochrome P450 contain heme in their structure. So all these Cytochrome P450 enzymes are heme containing enzymes, and they are considered as monooxygenase. Mono means single. So they add single atom of oxygen to the substrate. The substrate already contains H hydrogen atom. So on adding single atom of oxygen, it produces OH. That's why it is considered as hydroxylation reaction. So this enzyme belongs to monooxygenase enzymes. And when exposed to carbon monoxide, this group of enzymes having maximum absorption at 450 nanometer. That's why they are considered as cytochrome P450 enzyme. And this enzyme require NADPH as the coenzyme molecule. So the coenzyme molecule is NADPH. So coming to the phospholipid component of cytochrome P450, the major phospholipid or the lipid component is phosphatidylcholine. And all these enzymes are inducible enzymes, which means this can be activated by the effect of some drugs like phenobarbital group of enzymes can activate this cytochrome P450 enzymes. And their major role is concerned with detoxification. But cytochrome P450 is also involved in the synthetic function, that is synthesis of cholesterol and steroid hormone. So coming to the location where these enzymes are present in our body. So in our body, the detoxification function is predominantly done by liver. So cytochrome P450 enzyme is present inside the liver cell, and within the liver cell they are present in the smooth endoplasmic reticulum. So cytochrome P450 enzymes present in the smooth endoplasmic reticulums of the hepatocytes are concerned with detoxification reaction. And these enzymes are also present in adrenal gland. And in adrenal gland, these groups of enzymes are concerned with cholesterol biosynthesis and steroid hormone biosynthesis. And in adrenal gland, these enzymes are present inside the mitochondria. So they are present predominantly in the liver cell and also present in the adrenal gland. And in liver, they are concerned with detoxification reaction. And in adrenal gland, they are concerned with cholesterol and steroid hormone biosynthesis. So that's all regarding cytochrome P450. And cytochrome P450 is an important short answer question. So hydroxylation reaction is the major reaction in phase 1 metabolism. Coming to the other reactions of phase 1 metabolism, the next one is oxidation reaction. The classical example is oxidation of metabolism of the alcohol. So all of you know regarding the metabolism of ethanol. Ethanol undergo oxidation reaction and it is converted into acetaldehyde. And this acetaldehyde again undergo oxidation and it is converted into acetic acid. So for oxidation reaction, the best example is metabolism of ethanol. Ethanol, it is converted into acetaldehyde, and this acetaldehyde, it is converted into acetic acid. Other example is oxidation of benzene. Benzene on oxidation produces their phenol derivative. Coming to the next reaction in phase 1 metabolism, that is reduction reaction. And reduction reaction is important for compound containing nitro group. So the best example is conversion of nitrobenzene. So on reduction, this nitro group is converted into NH2. So nitrobenzene is converted into aniline. So reduction reaction is important for the chemical compound containing NO2 group. And the next reaction in phase 1 metabolism is hydrolysis. So the best example is the hydrolysis of aspirin. Aspirin is an antiplatelet drug. So on hydrolysis, this aspirin is converted into salicylic acid and acetic acid. Another example is hydrolysis of acetanilide. On hydrolysis, this acetanilide is converted into aniline and acetic acid. So that's complete phase 1 metabolism. So in phase 1 metabolism, the major reaction is hydroxylation reaction, and it is mediated by cytochrome P450 enzyme system. Other reactions in phase 1 metabolisms are oxidation, reduction, and hydrolysis. Oxidation reaction is important for the metabolism of ethanol and benzene. Reduction is important for the chemical compound containing nitro group. So that completes the phase 1 metabolism. So this phase 1 metabolism produces phase 1 metabolite, and this phase 1 metabolite undergo further metabolism in phase 2 reaction, that is conjugation reaction. So in conjugation reaction, this phase 1 metabolite undergo conjugation with water soluble compound or hydrophilic compound, so that this phase 1 metabolites are converted into more water soluble form, that is phase 2 reaction. So phase 2 reaction is also known as conjugation reaction. Phase 2 reaction is also known as conjugation reaction. So it can be conjugation with glucuronic acid, conjugation with glutathione, conjugation with glycine or glutamine, or it can be sulfation reaction, acetylation reaction, or methylation reaction. So phase 2 reaction is also another important short answer question. Phase 2 reaction or conjugation reaction. So if the question is conjugation reaction, you have to mention about conjugation with glucuronic acid, glutathione, glycine, glutamine, sulfation, acetylation, methylation with examples. So in phase 2 reaction, the phase 1 metabolite undergo conjugation with water soluble compound, so that they are converted into hydrophilic compound, so that they can be easily excreted from our body. Coming to the conjugating agent and their active form. In case of glucuronide conjugation, the active form of or glucuronic acid donor is UDP glucuronic acid. So UDP glucuronic acid is the glucuronic acid donor in glucuronide conjugation. And in sulfation reaction, phosphoadenosine phosphosulfate is the sulfate group donor. And in acetylation reaction, acetyl-CoA is the acetyl group donor. And in methylation reaction, S-adenosyl methionine is the methyl group donor. And regarding glutathione reaction, the major active group in glutathione is the cysteine residue. Coming to the glucuronic acid conjugation, the best example is the conjugation of bilirubin. So bilirubin on conjugation produces bilirubin monoglucuronide, and this monoglucuronide further undergo conjugation and produces bilirubin diglucuronide. So water insoluble bilirubin is converted into water soluble bilirubin diglucuronide, and it is mediated by glucuronide transferase enzyme. Coming to the reaction or detoxification by glutathione. And regarding glutathione, all of you know glutathione is a tripeptide. Tripeptide means it is made of 3 amino acids. So the amino acid residues are glutamic acid, cysteine, and glycine. And glutathione is also known as gamma glutamyl cysteine glycine. And regarding cysteine, all of you know it is a sulfur containing amino acid. Regarding the active group of glutathione, the active group of glutathione is the SH group present in the cysteine. This SH group is also known as sulfhydryl group. So the active group of glutathione is the sulfhydryl group of the cysteine residue. That is the active group of glutathione. Coming to the detoxification reaction of glycine, the best example is detoxification of benzoic acid. So glycine converts this benzoic acid into non-toxic hippuric acid. That is the example for detoxification reaction of glycine, benzoic acid into hippuric acid. Coming to the conjugation with glutamine, phenyl acetic acid undergo conjugation reaction with glutamine produces non-toxic phenyl acetyl glutamine. So that is the example for conjugation reaction with glutamine. Coming to the acetylation reaction, in acetylation reaction, acetyl group is added to this substrate, and acetyl group is donated by acetyl-CoA. So in acetylation reaction, acetyl group donor is the acetyl-CoA, and the enzyme which mediates this acetylation reaction is known as acetyl transferase. And this acetylation reaction is important for the detoxification of drugs like sulfanilamide and isoniazid. So drugs like sulfanilamide and isoniazid require acetylation reaction. Coming to the methylation reaction. In methylation reaction, the methyl group donor is S-adenosyl methionine, and the enzyme which mediates this reaction is known as methyl transferase. The best example is the metabolism of epinephrine. Epinephrine undergoes further catabolism and it is converted into metanephrine, and it is mediated by catechol O methyl transferase. So epinephrine to metanephrine is a reaction mediated by methylation. Coming to the sulfation reaction. In sulfation reaction, the sulfate group is donated by phosphoadenosine phosphosulfate, and the enzyme is sulfotransferase. So for sulfation reaction, the sulfate group is donated by phosphoadenosine phosphosulfate, And for methylation reaction, methyl group is donated by S-adenosine methionine. And for acetylation reaction, acetyl group is donated by acetyl-CoA. So that's all regarding phase 2 metabolism. So coming to the comparison of phase 1 and phase 2 metabolism, the major reaction in phase 1 is hydroxylation. So hydroxylation is the major reaction in phase 1 metabolism. Other reactions are oxidation reaction, reduction reaction, and hydroxylation reaction. And phase 1 metabolism produces phase 1 metabolite, and this phase 1 metabolites are less toxic compared to their precursor molecule or their parent molecule. And this phase 1 metabolite undergo further reaction in phase 2 metabolism. And in phase 2 metabolism, some hydrophilic compounds undergo conjugation with this phase 1 metabolism, so that it is converted into non-toxic water-soluble form, so that they can be easily excreted from our body. So phase 2 metabolism, the main reaction is conjugation reaction. There is one more detoxification reaction for this xenobiotic compound, that is phase 3 reaction. Regarding phase 3 reaction, it is an uncommon pathway. And in phase 3 reaction, the phase 1 metabolite or phase 2 metabolite, they undergo conjugation reaction with glutathione. So that's all regarding the metabolism of xenobiotics. So let's see some important questions. So the first question is phase 1 xenobiotic metabolism. So in this question you have to mention about different reactions seen in phase 1 metabolism, that is hydroxylation reaction, oxidation, reduction, and hydrolysis with examples. The second question is phase 2 xenobiotic metabolism or conjugation reaction. Here you have to mention about different conjugation reactions seen in phase 2 metabolism, that is conjugation reaction with glucuronic acid, glutamine, glycine, and glutathione. And you have to mention about acetylation reaction, methylation reaction, sulfation reaction is also seen in phase 2 metabolism. And the third question, that is cytochrome P450 enzyme system. So that's complete metabolism of xenobiotics. Okay, thank you.